In this video, I'll be discussing EEGs and fMRIs, which are brain imaging tools or techniques. EEG stands for an electroencephalogram. fMRI stands for functional magnetic resonance imaging. So in this video, I'll be contrasting them, letting first the EEG. The EEG technique makes use of electrodes to measure the brain activity or electrical activity from cells under the scalp. So it measures electrical activity from the cell population under the scalp, and it's really good for temporal resolution, which basically means that it can tell you down to millimeter precision when an event took place. In essence, we can talk about evoked potentials, which is the brain's activity in response to a certain stimulus, and an EEG is able to tell you that with really great precision as to when an event happened. But it is not very good at spatial resolution, as it is not good to tell you where the event happened, because the electrodes are all over the scalp, so it can't really differentiate as to what particular part of the brain and events that happened took place in. The fMRI stands for functional magnetic resonance imaging, and it functions by detecting amount of oxygenated blood or blood flow to a certain part of the brain. So it detects hemoglobin amounts in the blood in the brain. fMRIs are really good in giving high resolution images. But they also need a baseline, in essence something to compare and contrast the picture with. So they're good at telling you where an event took place. In essence, if a certain change happened in the brain, an fMRI is gonna be able to tell you where exactly that event occurred, but it's not gonna be able to tell you when. Because it works by comparing and contrasting two different pictures, so it can't really tell you with great precision as to when an event happened, but it is good at telling you where. And this is also a reason why it's not as good at temporal resolution, is also because blood flow is slower. So these are the two major more important brain imaging techniques available.